Good morning, everyone.
Good morning.
And welcome to the New Zealand Festival, the Enterprises Week. I don't know why you people are here. Shouldn't you be in church or at the beach or something like that? But welcome to this, what promises to be a very interesting discussion about the new age of genomics, what DNA, our genes and our genomes mean for health, for medicine, for society, for evolution, for the law, for everything really. So it's an incredibly important topic that's emerged in the last 10 years or so, how it is changing the face of science. I've got an amazing panel of scientists from around New Zealand with me. I'm going to get them to introduce themselves in just a minute. My name is Rutherford, and I'm a guest in the country. I'm from London, and I'm from the BBC. I present Inside Science, which is Radio 4's weekly science show, and I'm also a recovering geneticist, is how I describe myself, so I have a vested interest in this subject. Some housekeeping, so I have to ask you to make sure your phones are off or on silent. And just a few thank yous on behalf of the festival. I'd like to thank the Lion Foundation, Victoria University, Creative New Zealand, QT Museum, Wellington, Unity Books, and for their support of this session in particular, the Royal Society of New Zealand, the Dodd Wall Centre, Genetics Otago, MedTech Core, NZ Genomics Limited, and Te Punaha Matatini. So, just another note on housekeeping. We're going to have a nice, relaxed conversation for half of the session or so, and then we're going to raise the lights, because this is a dialogue between the scientists and the audience. So you're welcome to ask questions. When it comes to questions, if you could put your hand up very clearly, there are microphones, so we have volunteers with microphones, and I will direct a question to you. Wait for the mic to come to you. I've got 3 simple rules for questions in sessions like this. One is, please make it a question, not a statement. The second is, please make it a short question, so we can get as many questions in as possible. And the third is, please try and be sane, if that's okay. I've been in many sessions where that third rule hasn't really transpired, but you look like a very sane audience. That's all the introduction from us. So what I'm going to do now is I'm going to ask the panel to introduce themselves, and then we'll get going. So perhaps Peter, if you want to start.
Do it stand up?
No, don't stand up.
I normally talk standing up, so this is a bit strange. And also the lights in my eyes. Are you still there? Yeah, good, he's there. Excellent. So my name is Peter Dearden. I am Director of Genetics Otago. So that's a research centre of the University of Otago. My research is varied. I'm an insect geneticist, mainly studying how embryos evolve, so looking at how genes control how an embryo is made, and then how those processes change to give you different forms in evolutionary biology. But I also do a lot of work for the beekeeping industry, so I look after the genetics of bees and do selective breeding in bees, and is that enough?
It is, and just in case you're interested, genetics is a very broad description of a huge area of science. Basically, it encompasses all biology. What Peter just described, which is how genetics affects development of the embryo, is factually the most interesting bit of all biology, and I can say that because that's what my PhD is.
Absolutely true, and I'd like to support. Move on.
But there are other aspects to biological science.
Thank you so much. So I'm Susan Morton. I'm a life course epidemiologist, so rather than looking at the molecular level, I'm much more looking at the population level of things. So I am a public health physician, so I'm a doctor by training. I'm interested in solving wicked problems for our population and trying to understand why we see such vast inequalities in outcomes, particularly health outcomes for our population. I'm also by training a mathematician, so I'm rather a strange geek who likes to understand patterns in outcomes, and that's where I apply that to the population that I'm looking at. Currently, I'm at the University of Auckland, where I'm the director of the Centre for Longitudinal Research, and I have directed the new longitudinal study of New Zealand children and families called Growing Up in New Zealand since its inception in 2005.
And for the record, epidemiology and transgenerational longitudinal studies are the most interesting bit about medicine. So, honestly, this panel is hand-picked.
Thanks, Adam. So my name is Colin Gavaghan. This is not violent, as I should say, this is not a Dunedin accent. I'm from Scotland originally, I've been here a few years now. And I'm not any kind of scientist at all. Didn't even get a no grade biology at school. What I am is a lawyer, and I'm one of those fun people that likes to come along to panels where scientists are talking about all the cool, exciting things they're doing, and to tell them why they can't be doing it or shouldn't be doing it. So I'm the director of the New Zealand Law Foundation Centre for Law and Policy in Emerging Technologies, which is a centre in desperate need of a catchy acronym. And that's based at the University of Otago, and we explore the kind of interactions between law and policy, obviously, and emerging technologies, with a particular interest in genetic and reproductive technologies.
Okay, thank you, panel. So I want to start off by we are talking about an era of biology which was really kicked off in sort of 2001 or 2003 when the first human genome was published, and that was, what, 13, 14 years ago now. And so really, what we're talking about is how that publication, that advance in science, that grand project, how that has fundamentally changed pretty much all aspects of biology, and as a result of medicine, or many aspects of medicine, and also society, and as Colin was saying, the law. So I want to start by going down the panel and just asking a relatively simple question, although I expect the answers are quite complex. And I'll start with you, Peter. How has the genomic age changed the work that you're fundamentally interested in?
Yeah, so this is interesting. When I was a student, I was asked to become, I got a job, which I turned down as mapping the alcohol dehydrogenase locus of Drosophila melanogaster, which is a fruit fly. So that's mapping a really boring gene in a really quite dull organism. And I turned it down because it sounded dull, but what it was in fact the start of the Drosophila genome project, which was sequencing all the genome of this fruit fly, which is genetics' favourite fruit fly. And that was as a test to see whether we could use the same techniques to sequence the human genome. So I skipped out on that, and then I spent a lot of time saying that the Human Genome Project was a load of crap, because while we could sequence the human genome, we would never understand it. There's so much information in there, and our tools for understanding what genes do generally involve breaking them, which is hard to do in humans. Well, it's not hard to do, it's probably not ethically acceptable.
Can we just check the lawyer on the panel?
Okay, so that's good, but that's weaker than I usually get that. So, but then as the Human Genome Project went along, it developed a whole load of technologies and ideas, and in fact, when it came out, it was a real triumph. And I don't think I'm overstating it, that this is as important scientific advance as putting people on the moon, right? It's a really huge thing. We now have the sequence of the human genome, and that means for me that actually the capacity of all the workers and the genome sequencing machines that were used on the human genome suddenly became free for me to play with. And so soon after the human genome was sequenced, we started sequencing the honeybee genome, which is far more interesting than the human genome, because we can break genes in honeybees. Yeah, good, we're right there. We can and learn what they do. Of course, honeybees are the most important insects on Earth. The same applies. So actually, the human genome was kind of a start of a wave of technologies and a wave of genome sequencing, which has changed the way that I do everything. So in the past, we used to guess what genes were involved in a particular process. Now we use modern high-throughput sequencing to identify the genes by saying, okay, well, these genes are expressed differently. We can now sequence, in a very short space of time, all of the genes of an organism, or all the genes which are turned on or off at a particular time. And that's just transformed the way that I do my work. I guess what happened after the Human Genome Project was that new technologies came on board, which meant that we could sequence genomes much faster, much more cheaply. So the human genome cost $3 billion, I seem to remember, and it took 15 years, and millions of people, well, hundreds of scientists around the world were involved in it. Now I could sequence your genome in a day and a half for less than $1,000. That change in technology has opened up whole new areas of genetics, which frankly we need to talk about.
Sure, and I think the fields of evolutionary biology and how genetics relates to evolutionary biology are perhaps the most obvious examples of a very clear relationship between progress and the Human Genome Project and those technologies you just described. But I guess in medicine, Susan, those links are not quite as clear. I remember Bill Clinton standing on stage in 2000 and saying that within a few months we would have identified every gene causing a disease, and it won't be long before we would have cured them all.
Exactly.
How many diseases have we cured as a direct result of the Human Genome Project?
That'll be a big zero, actually, unfortunately. So yes, I recall that as well, and I recall being sceptical about it at the time. I think there's no doubt, as Peter has said, that the sequencing of the human genome has been an amazing advance in biology. But when you're looking at it from a population perspective, trying to understand what causes health and well-being and diseases in a group of people that we tend to call populations, whether those are country-specific or subpopulations in countries, then you really need to understand their biology, but also their environment, or if you like, nature and nurture. And of course, while we now know how complex that nature is, the genome, and it is remarkably complex, and I think that's been stunning to find out, to sort of break the idea that we had one gene, one protein, and we have this whole sort of amazing sort of interaction of DNA that is shaping all of the things that are possible for a human. But what we still don't understand yet in terms of solving disease, if you like, or the cause of disease, is how that sort of unique pattern intersects and interacts continually over time with the environments that an individual is exposed to and the other individuals that that one individual is exposed to. So Peter has said quite rightly, if I gave him $1,000, he could tell me what my sequenced DNA was. But I haven't got $1,000, and I haven't got $1,000 for the 7,000 children who are in the Growing Up in New Zealand study. And that's sort of the issue, that yes, it's cheaper, and I hope one day it will be cheaper still, because what we're doing with the 7,000 children, that we are interested in understanding what shapes every aspect of their development so that we can start to really make a difference to those children's well-being. We do want to understand how their biology, complex or not, interacts with the environments they are exposed to, even from before they are born, even across generations. And how do we therefore look at where we intervene? How do we make change to change the trajectories of development of those children? And for me, I guess, as a doctor, as an epidemiologist, I see the biggest bang for our buck actually being at the environmental level, finding those environmental conditions that are modifiable, that will allow us to actually work with the genomes that we're born with, all of us, and actually understand how we can get the most potential out of those. And I guess that's where I see the potential, but I see it as a beginning. I see that the sequencing of the genome is an amazing feat, but it's a beginning of understanding disease causation, of prevention, of treatment. And I think that's exciting, and I think it's exciting that we're actually working across disciplines and from the molecular to the population level to try and solve those problems.
I mentioned this in the session we did yesterday when we talked about some of the sort of similar issues, but one of the quotes that was given at the time, actually a little bit after, was, and apologies if anyone was in that session because I'm just going to repeat myself, but one of the leaders of genomics nowadays has referred to the human genome as being Rumsfeldian. So that great philosopher of science, Donald Rumsfeld, he was referring to the weapons of mass destruction in Iraq, in the first Iraq War. He said a very clunky, clumsy thing, which was there are known unknowns, there are unknown unknowns, and there are known knowns. And in fact, you know, it sounds ridiculous, but in fact, when in referring to genomics, the Human Genome Project was the transition from unknown unknowns to known unknowns. So after the genome was completed for the first time, we began to see the landscape ahead of us of what work needed to be done in order to understand human diversity. Colin, I think from a legal and ethical point of view, it also has had profound implications for medicine, for the way we think about biotechnology, and there have been massive ownership issues, haven't there?
Yeah, absolutely, Adam. I think if I were to characterise the relationship between genomics and law, we can probably see two strands emerging.
Can you just move a bit towards your hand, sister?
That better?
Yep.
That better?
Yeah, good. So on the one hand, we can see the use of the relationship as being one of a target for the law, a regulatory target. How should the law respond to the kinds of choices that genomic information make possible? So my own